Let's dive right into this explainer. Today we're tracking the epic microscopic journey of a single cell as it prepares, grows, and ultimately divides to create life. It's honestly one of the most fundamental yet completely mind-bending processes in all of biology, the cell cycle. It's a perfectly choreographed sequence of events that literally every living organism relies on to exist.
To kick things off, we really have to ask ourselves a pretty staggering question. How does a single, incredibly tiny entity contain the entire blueprint to build, operate, and heal a complex organism? I mean, you are made up of several billion cells, organized into over 200 major types, but all of that complexity, every single specialized function, originates from the division of prior cells. The sheer mechanics of how a cell safely copies its massive instruction manual and physically splits in two is just an absolute engineering marvel.
So here's our agenda for today. First, we'll look at the blueprint of life. Second, interphase, preparation and growth. Third, mitosis, the great divide. Fourth, meiosis, the reduction division. And finally, we'll wrap up with why cell division matters.
All right, Section 1, the blueprint of life.
Before a cell can divide, it has to know exactly what it's dividing, right? It needs a manual. To do this, it first has to organize its messy room of genetic yarn, which we call chromatin. Chromatin is essentially this complex network of super long DNA molecules packaged by special proteins. And this packaging is absolutely vital. It's what allows incredibly long strands of DNA to actually fit neatly inside a microscopic cell nucleus. To put that into perspective for you, if you stretched it all out, the DNA in a single cell would be about 2 meters long. But during its normal day-to-day life, this chromatin is pretty loose and uncoiled. Think of it kind of like a tangled bowl of microscopic spaghetti.
But when the cell actually gets ready to divide, that loose chromatin condenses into these highly structured, robust chromosomes. As division kicks off, each chromosome consists of 2 genetically identical copies of threads. We call these sister chromatids. Now, these identical sister chromatids are joined securely together at a central point, known as the centromere. This whole condensed structure is absolutely crucial because it ensures the DNA blueprint can be safely and accurately passed on to daughter cells without getting tangled up or broken.
Moving on to Section 2, interphase, which is all about preparation and growth.
With our DNA neatly packaged up, our protagonist cell enters the first major phase of its life cycle. Now, historically, people thought of interphase as a resting phase, but it's actually an incredibly active factory assembly line. It's broken down into 3 distinct subphases. First up is the G1, or gap 1 phase. This is a period of massive metabolic activity. The cell is physically growing in size, synthesizing specific enzymes, and crucially, accumulating the DNA base units it's going to need later. Next, we hit the S phase, or synthesis phase. This is the real magic moment where DNA replication actually happens. The chromatin material gets duplicated, so we end up with 2 exact copies. Finally, the cell enters the G2, or gap 2 phase. This is the pre-mitotic phase where the cell grows even more, replicates its organelles, and synthesizes the final enzymes required for the big upcoming division.
Now, a crucial detail here is that not all cells are just constantly rushing to divide. Sometimes, at a specific point during that G1 phase, certain cells hit the biological pause button. They detour into what's known as the G0 or G0 phase. In this state, the cell cycle just stops. The cell goes about its normal functional life, but it doesn't prepare to divide. It might hang out in G0 for days, weeks, or in some cases, like with certain nerve cells, literally for your entire lifetime.
All right, Section 3, mitosis, the great divide.
For the cells that do push through G2, they are fully prepped and facing their climax, the M phase, or specifically mitosis. After the initial prophase where the nuclear membrane breaks down, we land at metaphase. Look at how those condensed chromosomes arrange themselves perfectly along the equator, literally the absolute middle of the cell. Meanwhile, spindle fibers, which were formed by centrioles at opposite ends of the cell, attach themselves right to the centromere of each individual chromosome. It creates this intense biological tug-of-war that is perfectly balanced right in the center.
Then, instantly, the cell shifts into anaphase. The tension breaks, the spindle fibers contract, the centromere divides, and those sister chromatids are violently pulled apart. Each chromatid, which is now officially considered its own independent chromosome, gets dragged to the exact opposite poles of the cell. So one complete set goes to the left, and the other complete set goes straight to the right.
Once they reach the poles, that marks the beginning of telophase. The frantic movement finally ceases. Now, each pole has the exact same number of chromosomes as the original parent cell. Those chromosomes start to uncoil back into that loose chromatin state we talked about earlier, and a brand new nuclear membrane reforms around each set. So we now have a single cell containing two completely identical nuclei. Finally, to finish the whole job, the cell undergoes cytokinesis. The cytoplasm itself physically pinches in half. In animal cells, you get this constriction that develops and deepens until it literally cuts the cell in two, whereas in plant cells, a cell plate actually forms down the middle. But either way, this successfully concludes our hero's journey by birthing two identical daughter cells. These exact copies now enter their very own interphase, and the entire cycle starts all over again.
Which brings us to Section 4, meiosis, also known as the reduction division.
So mitosis is amazing for everyday growth and repair, right? But what if the goal isn't just to make a copy, but to create new life entirely? Well, that requires a completely different set of rules. Meiosis is a highly specialized series of two divisions. In meiosis I, the first meiotic division, the cell takes a different path. Instead of just lining up individual chromosomes, it pairs up matching, or homologous chromosomes. When the cell divides this first time, it separates those pairs. We call this a reduction division because it essentially cuts the total amount of genetic material right in half.
Next up is meiosis II. After meiosis I, there's a short interphase, but crucially, the DNA does not replicate again. The cells immediately plunge into a second rapid division. The chromatids separate, a lot like they do in mitosis, but because the DNA wasn't duplicated first, the ultimate result is 4 unique haploid cells. These are cells with only half the normal number of chromosomes, making them perfectly primed for sexual reproduction.
Let's put these two epic conclusions side by side so we can really lock this in. Mitosis involves one division to create 2 exact copy daughter cells. That's the process used by your somatic or everyday body cells. Meiosis, on the flip side, involves 2 divisions to create 4 totally unique haploid cells with half the chromosomes. That process is strictly reserved for reproductive cells. Basically, mitosis maintains the status quo, while meiosis is what drives genetic diversity.
Section 5. Why cell division matters. We've looked at all the microscopic mechanics, the spindles, the phases, the chromosomes, but why does this actually matter to you and me right now? Well, these exact cellular cycles are constantly working in the background of your life. Every time you scrape your knee, mitosis is what steps in to heal that wound, repairing the wear and tear. It's the reason you grew from a single fertilized egg into the complex person you are today. It's constantly producing your vital new somatic cells, like replacing your red blood cells day in and day out. And in the broader natural world, it even allows for asexual reproduction and vegetative propagation in plants. It is quite literally the engine of biological maintenance.
It's truly mind-blowing when you think about it. Just while we've been sitting here running through this explainer, millions upon millions of your own cells have successfully completed this exact cycle. They unpacked their chromatin, replicated their DNA, lined up perfectly on their microscopic equators, and divided, all just to keep you alive and functioning. It really leaves us with a profound appreciation for the invisible biological factories constantly working inside of us. So, how many cells do you think is divided in your body over the last 10 minutes? Thanks for joining me, and keep wondering about the incredible science happening right inside of you.